people have trouble with. All right, so let's go over this. Gave you a little bit of background information that we talked about, also about ender and exergonic reactions, and it asked you for the symbol of the change in the entropy, which is delta S, right? And then it asks you when the entropy of a system increases, what sign, positive or negative, is used. And so delta S is really S final minus S initial, right? And so if it increases, if the entropy increases, that means the final, you're going to have more entropy than your initial. So therefore, when you subtract that, you're going to have a higher number subtracting a lower number. And so we're going to get a positive for that. Number 2, is there a correlation between the level of organization of molecules and a change in entropy, and justify your reasoning? So, so think about this. When you have an exergonic reaction, like the gasoline example I gave you yesterday. So you have this exergonic reaction where you get a lot of energy released when we break down gasoline. Has the entropy of the universe changed as a result of burning that gasoline? Yes. And what has happened to organization? You've had gasoline, which had all this energy stabilized as chemical energy in the gasoline molecules. We broke those molecules, those gasoline molecules down and reformed them into molecules that have less energy, and that's why energy is released. And so therefore, does the organization play a role in the amount of entropy? Absolutely. So when you burn the gasoline, the products have less free energy, but we have more entropy in the universe as a result of that. All right, and so therefore, the answer there is yes. Okay, and so then we look at number 3. This is all about entropy here. It says predicted the following processes would have an increase or decrease in entropy based upon what you've learned. And so if we look at here, water evaporating from a wet sidewalk. Water evaporating from a wet sidewalk, we have an increase in entropy. Increase in entropy, why is that? You might have an explanation about why that would be. Madeline?
Because it's like changing state of matter, and so when there's a chemical change like that, some of like the energy is released as heat energy.
Okay, all right, yeah, absolutely. And so then when the water evaporates, we also have the water as a liquid is more organized. All right, they're closer together. Remember, they're moving less, and so then when they're evaporate into the atmosphere, they are more disorganized as well. All right, and so increase in entropy. Then a plant using raw materials to build a leaf. This with entropy, what kind of reaction would that be? Exer or endergonic, do you think? Plant using raw materials to build a leaf in the spring. Exer or endergonic? Megan? What was that? Endergonic, right. And you need to have an input of energy in order to do that. And so what happens to entropy then if they're having an input of energy to build these molecules within the leaf? Go ahead, Megan.
Decrease in entropy.
A what?
Decrease.
Yeah, decrease. And here it's absorbing energy from the atmosphere. Alrighty. Then it talks about spontaneous reactions and law of thermodynamics and so on. And then it asks you predict the change in entropy of the surroundings for an exergonic reaction. And this is, so the change in entropy of the surroundings for an exergonic reaction, this is an increase. Because remember, an exergonic reaction is going to release energy. You go from molecules, remember this graph here, like here's reactants and products, and you get energy released. And so what happens is the energy released, the energy was once stored as chemical energy, and now some of that could be released as heat energy, which is more disorderly. And so here we have an increase of entropy from at the final state. So then on the flip side, the change in entropy of the surroundings for an endergonic reaction, that would be, alright, for this same reason. Because look, when you look at this, an endergonic reaction absorbs energy, so it's gonna take energy like heat energy and put it into more stable energy in the chemical energy of the bond for the product. Alright, does that make sense? Okay? Alright, flip it over. And it talks about the equation here, and then we have to apply this. So a polypeptide chain is converted to individual amino acids. So we're breaking down a molecule, long chain of amino acids to individual amino acids. And then it says, would the delta H be positive or negative? The answer there is negative. Let's talk about why that is. So when we talk about, figure out the delta H is H final minus H initial. So when we think about this type of reaction where we're breaking down the molecule into individual amino acids, is that an exergonic or endergonic reaction, do you think? Exergonic, and so it's going to release energy, and so the polypeptide chain is going to have more free energy than the individual amino acids at the end, so therefore at that difference there, that is going to be energy that's going to be released. And so if we look at H final and H initial, the final is going to be less than the initial. There's more total energy initially than in the final state. So therefore, when we take a lower number and subtract a higher number, we get a negative number. So that's why I get the negative number there. So then B, would the delta S be positive or negative? And that is positive. And so let's talk about why that is. So if we do the delta S, well, we can just do it right here. Delta S is S final minus S initial. So when we talk about entropy, the disorder, there is more disorder at the end in the final state because all of that chemical energy that was once stored in these molecules, some of that energy is still chemical energy in the bond, in the products, but a lot of that chemical energy has been converted to a different form of energy, and that's been released, and that could be heat, which increases the disorder. And so therefore, your final entropy is going to be higher than your initial energy. So when you take a higher number minus a lower number, you get a positive number. And then C, show how the change in enthalpy and entropy of the above process would result in a spontaneous reaction at all temperatures. So then this is looking at the delta G equals delta H minus T delta S. And so we just talked about how the delta H would be a negative number. Delta S would be a positive number. So we're going to take this negative number and subtract a positive number, which means that you're always going to get a negative number, which means that energy is released. Remember that the negative means in front of something, like yesterday we did a couple of examples where, you had to figure out if it was positive or negative, the delta G. Negative, if we have a negative 7 kilocalories per mole, let's say, this is the amount of energy. What this means is that energy is released. That's all the negative or positive means. It tells you if the energy is released or is needed. The number tells you how much. Does that make sense? All right. And so therefore, you're always going to, an exergonic reaction like this, you're always going to have a negative number. In an exergonic reaction, you're going to get energy released every time. Okay. And so then, when you look at number 7, we have this process here. In a not a spontaneous reaction, this process here is what process? Do you guys remember what this is? I put it on the board and talked about it. Yeah, this is photosynthesis, making glucose. So we're making glucose from carbon dioxide and water, and we also have oxygen that's released. So, let's just talk about this photosynthesis. Is photosynthesis an exergonic process? Does it need energy or does it release energy? It needs energy, right? And so in this case here, your carbon dioxide and water have less free energy than your end products. Glucose has way more energy in it. So your graph looks like this. So that difference, you have molecules that have a ton more energy in them than your starting molecules. So that difference means that energy is needed in this case, which is an endergonic reaction. So it's asking you what's the entropy change for the system. Is there an increase or decrease in entropy? So when we look at entropy, that's disorder. And so looking at this, is there more disorder initially, in the initial state, or the final state? So we're really talking about delta S. Is it an increase or a decrease? So final minus initial here. So there is actually more disorder in the initial state here because this energy could be heat energy that's absorbed into the system and is now stored in these products. And so by making these products, there's less entropy. And so for the initial state, there's more than the final state. And so therefore, as we go from initial to final, that would be decrease, right? Okay. And then this one here, because this is asking about the system, I'm gonna just, it says what's the entropy change for the surroundings? I will not ask, this is kind of above and beyond, I forgot to tell you not to do this question, above and beyond this class. But what do you guys think? What do you guys put for that? Megan?
Increase.
Increase why?
Because, like, you're taking energy from the surrounding area, so if it's going to become organized, if it's becoming less disorderly, then the surrounding area is, like, less disorderly because it's taking into the system.
Okay, absolutely. All right. And so it is this, but like I said, for this class, it's more this type of stuff that you need to be able to do with that. Okay. All right. How'd you guys do? Okay. Yeah. All right. I'm gonna try and come up with some more problems as practice for this, because this is something typically that a lot of kids have trouble with. All right. All right. All right. So we're going to continue on in our notes. So what we're going to do today, we're going to do a little bit of notes with talking about enzymes. Although we have to do a little bit more in here. We leave off here.
Yeah.
Talk about this picture.
Yeah.
Right after that.
But this is where we're at.
Yeah.
And speaking of which, when we've got, we just went over this, and so on, I wanted to just give you a heads up of what I'm thinking of starting and trying something with next chapter, which is chapter 9. This is my thoughts, because I feel like a lot of the learning takes place in doing stuff like this and applying what we've talked about and so on. And so this is what I'm going to try for chapter 9, and we'll see how it goes. It's like a little experiment, all right? What I'm going to do is I'm going to flip what we do. So on my conference hour, my thought is on my conference hour, I'm going to videotape myself with notes. Your homework is going to be to watch and take the notes. And then in class, we're going to do stuff like this where we apply the stuff in the notes so you can talk amongst yourself to learn better and can ask me questions and so on. I think that, because I think you can do the notes by yourself, but I think talking and having conversations with people about the content and having an opportunity to ask questions right when you think of them when you're doing them will enrich your learning experience. All right, so I thought I would try that out. What do you guys think? Games? And then we can evaluate afterwards and see how we liked it, if you really thought it was better or worse or whatever. But just thought I'd try it out because I feel like there's a lot of people that could benefit from getting some extra help, and quite honestly, not a lot of you are coming in for that extra help. So I'm thinking that this might be a way where you're spending time at home and then also spending time here and doing that. So we'll try it out and see. All right, and so that's going to start next chapter. Okay, and we'll see how it goes. Yes.
Would it be like a full hour of notes like we do in class, or like how long?
It depends. But yeah, it might be like yeah. Because technically, the College Board asks, like they say that it's an average of an hour a night you should be spending. I don't think people are spending that much time on this class, and so I can say till I'm blue in the face that you should be spending time every night reading the chapter, looking over your notes. People aren't. I don't think a lot of people are doing that, and so this way you should be watching the video and at least spending some time, and then you're spending time in class too. And so it's my way of like forcing you to like spend some time with it. Yes, absolutely. Nice choice of words. I was gonna say that, but yes, yeah. So that's the idea. So we'll try it out and see. I've been talking about this with Mr. Burdick across the hall for a long time. Him and I have been doing the videotaping for a couple years now, and he actually started flipping the classroom about a week and a half ago.
Sorry for the interruption. Thank you.
All right, thanks. He started flipping the classroom about a week and a half ago, and his students, and a lot of parents said he's gotten, because we just had parent teacher conferences, he's gotten really good feedback about how it's working out. So I thought it would be worth a shot for us. All right. So that's just a heads up on kind of what's coming up.
So will that mean that more time in class to do anything?
Yeah, you're going to be doing stuff like this, like homework and stuff like that. So your homework would be to watch the video, and this is going to be done in class.
Okay.
Does that make sense?
We won't have like other homework.
Right. So you're going to have class time to do what you would have done at home. Okay? So that's why it's flipping. So we're still doing equal time, you know.
So when are you recording the video?
So my plan is, I didn't even look at it. Did it say now? Okay. So my plan is I would record, I'm going to use my conference hour, which is third hour, and upload it during the day.
And that would be the homework for that night, and then the next day we would do like what our normal homework assignment would be.
Yes.
Oh.
So you're gonna. So this, I think it frees me up to do time-wise a lot of like maybe simulations and different modeling things that help you to visualize and understand the material better. Because time-wise, I have a time crunch to get done by May 9th, or is it May 8th this year? May 8th, you know, this AP test, and so I can only spend so much time day-wise on certain materials. So I thought that it would allow me to do more enriching activities that would help you to understand the material better. Okay, that's the thought process. Okay. So what do we do after May 8th? I have some stuff. We still do stuff. So yeah. But we got. Okay, go ahead. You gotta get. Mm-hmm. All right, let's get going on our notes then. So I just want to give you a heads up on that. Alrighty. So if you recall, ATP, let's get going on here. So ATP is an exergonic process. When we break it down, it releases energy to do cellular work. And what was the reaction that what do we break down to get energy to regenerate ATP and make it again? Scott? Glucose. And so let's look at how ATP can drive chemical reactions. So here you have an endergonic reaction. Glutamic acid and ammonia bond together, and that is a endergonic reaction. So remember, the positive means that it needs energy. The number tells you how much energy is needed for this reaction. And did I talk about kilocalories yesterday in this class? All right, so it says kilocalories per mole. A kilocalorie is a unit of measurement of energy. So when we look at the back of our food labels and it says how many calories are in your food, that's actually kilocalories. A kilocalorie is a calorie that's spelled with a capital C because there's uppercase calories and there's lowercase calories, and the uppercase calories are 1,000 lowercase calories, and that's a kilocalorie. So this is just a measurement of energy. So that's what that's telling you. So this needs energy. Here's your breakdown of ATP into ADP and a phosphate. That's an exergonic reaction. It releases energy. So that's the negative telling you we're releasing energy. The 7.3 is how much energy is released to do work. And is this amount of energy enough to drive and power this endergonic reaction? Is this amount of energy enough to power this reaction? The answer is yes. Because remember the negative, this is not a negative number. This just means the negative means that the energy is released. So it's releasing how much? It's releasing 7.3 units of energy. This one only needs 3.4. So this is releasing enough energy to power this reaction, then some, all right? And so if we actually subtract this, we get an excess of 3.9 kilocalories per mole, because only 3.4 of these 7.3 is used to drive this reaction. And so realistically, this amount of energy from one ATP could actually drive and make 2 of these reactions happen. There's enough energy for 2 of those reactions to happen. And so what happens is that's how an exergonic reaction works. It has to release enough energy, more energy than the reaction needs. So what if you have a reaction, so here's your ATP molecule. Let's say you have a reaction that needs 300 kilocalories per mole. So that's positive 300. Then what that means is you need multiple ATP to do that, okay? So that's how that works. And so another way, at the beginning, yesterday we talked about the 3 processes or 3 types of cellular work: mechanical work, transport work, and chemical work. This is what we just looked at. This is the same reaction that we just looked at here, where this molecule here has a phosphate group on it. This wasn't shown, we didn't show that here. All right, this doesn't show the phosphate group on it. So this is showing you more specifically what ATP does to provide energy. So when ATP breaks down into ADP and a phosphate, that phosphate group actually attaches to a molecule. And this squiggly line means that this is an unstable bond. And what that does is allows this bond to be easily broken and these 2 to hook together. And so that's what we see here, and the phosphate is released. And so that's actually what the ATP is doing. Back on this here, when we talked about ATP releasing the phosphate and providing energy, that phosphate actually attaches to a molecule. We say this molecule is phosphorylated. It gets a phosphate attached to it, and then when the phosphate breaks off because of that unstable bond, it provides the energy for these guys to hook together. So that's chemical work. The 2 other types of work are mechanical and transport work. Let's talk about the transport work. Transport, what is this right here? You guys remember from chapter 7? Megan?
Is it a pump?
Yeah, it's a pump. So like the sodium potassium pump. Pumps, remember, to transport substances across the membrane. Pumps require energy in the form of ATP. Why does it require energy? It's moving. What, Ellie? Scott?
Against the concentration.
Against the concentration gradient, which means low to high. All right? And so what happens is, notice here, when we break down the ATP, so here's your ATPs needed, you break down the ADP to ADP and a phosphate, this membrane pump becomes phosphorylated, which then when it breaks off allows a shape change to allow the molecules to be transported across the membrane, and so that phosphate then comes off. And so when ATP breaks down into ADP and a phosphate, before it actually becomes ADP and a phosphate, that phosphate is attaching to something and then providing the energy to, in this case, change the shape of the membrane pump. And then mechanical work here. Remember when the cell unit, we talked about the cytoskeleton and the microtubules. And remember I said that the organelles can ride the microtubules like a subway, can move around the cell. And so that's what this is. So this would be like a microtubule. And this is a motor protein that becomes phosphorylated, and then notice when the phosphate group comes off, it provides energy for the protein to move across that microtubule and go from one place to another in the cell. So in the end, you end up with ADP and a phosphate, all right, at the end. And so, so we say that these molecules, in between here, the molecules, I'm going to write here, become phosphorylated. I want you to know that word. That means a phosphate group attaches and detaches. And then become dephosphorylated. So just so you know these words if you see them. Where the phosphate comes off. So that happens kind of in the middle of the ATP and the breaking down it breaks down into ADP and the phosphate group, but before it's just kind of hanging out, it attaches to a molecule. Okay? And that's how ATP works. So it's an exergonic reaction driving endergonic reactions. All right, so that's the first part of the chapter, which is just about the different types of reactions, exer and endergonic and so on. Now we're gonna come in and talk about how enzymes work with chemical reactions. So we all hopefully remember that enzymes do what to reactions? Speed them up, right? And so we're going to look at how they actually do that because it has to do with energy, all right, and so on. So enzymes are biological catalysts. Catalysts are a broad term for anything that changes the rate of reaction and speeds the reaction up. So in the universe, light can be a catalyst and speed a reaction up. Heat can be a catalyst. So enzymes are biological catalysts found in living cells. And so, so an example of heat and light with a catalyst, hydrogen peroxide, a bottle of hydrogen peroxide. We're all familiar with that. Hydrogen peroxide is used, we clean out cuts and so on. What happens is that's H2O2, and when hydrogen peroxide breaks down, it breaks down into water and oxygen gas. And so we actually have enzymes in our cells. And so what happens is called catalase. And what happens is when you cut yourself, you open up those cells and you get these enzymes exposed to the environment, and then you put hydrogen peroxide on it, and these enzymes break the hydrogen peroxide down into water and oxygen gas. So that's why it bubbles when you put hydrogen peroxide on it. It's actually the oxygen gas that's being released from these enzymes that are breaking down your hydrogen peroxide. So why the hydrogen peroxide is in a brown bottle? If you read the label of how to store hydrogen peroxide, it'll tell you a couple things. It'll tell you to store in a cool place, and the reason why is because heat is a catalyst. So catalase, the enzyme in our cells, is one a catalyst that can break down hydrogen peroxide, but heat is also. So if you expose it to heat, that'll speed up the breakdown of hydrogen peroxide into water and oxygen gas. And you don't want that. You want your bottle of hydrogen peroxide to stay hydrogen peroxide so that when you get cut, it's usable. The second thing, why it's in a brown bottle, is because light is a catalyst. So therefore, the brown bottle blocks the light, and therefore it stays as hydrogen peroxide. All right, so those are examples of non-biological catalysts, heat and light, and biological catalysts, enzymes. And so when I speed up metabolic reactions, Why? Because they lower what's called the activation energy. Activation energy is energy needed to start a reaction. So yesterday I gave you the example of gasoline, right? And remember that, I talked about that in this class, right? So you have the gasoline, gallon of gasoline, it is not going to automatically, when it comes into contact with oxygen, automatically combust and we're not going to have a big explosion. Why? Because we need to have an input of energy. That input of energy, even for exergonic reactions, are called activation energy. And so that's the input of energy, and then you get the big exergonic reaction, you get energy release. And so, so enzymes, what they do is lower the amount of energy. So if you don't need as much energy for the reaction to occur, then it can occur faster because it doesn't have to absorb as much energy, and therefore can occur quicker than waiting until it absorbs a higher amount of energy. So this is an example that I actually used in the chart that we went over yesterday. Sucrose being broken down into glucose and fructose. You guys remember if this should be a positive or negative delta G? I did this yesterday. Energy. This was the first example we used. So it is negative, yes. All right, and so, so building on that, here's your disaccharide, and hydrolysis is going to occur. So you add the OH and the H, so you get up with 2 monosaccharides. And notice here this sucrase. Anything, if you see ACE on the end of something, that refers to an enzyme. Now, if you see something with ACE at the end, that means it's an enzyme, but not all enzymes actually have ACE on them. So there are some exceptions with that. So notice here they try to name it with what it is speeding up. So sucrose, os remember means the sugar. So sucrase speeds up the breakdown of sucrose. All right, and so that's your enzyme. Also notice that your enzyme is written on the arrow. And so that means that it's not a reactant. It's not a reactant, and so therefore it's not going to be converted into a product and so on. This sucrase can break down sucrose, and then once it does that, that same enzyme can go and break down another sucrose and another, and so on and so forth. So let's look at this, how it lowers the activation energy. Let's look at this black line first. You're looking at the X and the Y axis are really the same as what we talked about yesterday with the bar graph. So this is time, progress of the reaction, this is the free energy. So your reactants, this is how much free energy in your reactants, and this is how much free energy in your products. So if we relate that to the bar graph from yesterday, your bar graph from yesterday, how that would have looked like here is this is the amount of free energy in your reactants. So you would have had the bar go up to here. And then this is the amount of free energy in your products, and so the bar would go up to that dotted line. So that's how this relates to this. So this is the amount of free energy, reactants, and products. Is this reaction, before we even look at the lines, just looking at the free energy of the reactants and the products, is this reaction exer or endergonic? What am I looking at to tell that? Is this reaction exer or endergonic? Megan? This is exergonic. Is that what you're going to say, Madeline?
Yeah.
Can you tell me why?
Because the reactants seem to have like more free energy than the products, because when the reactants break down, some of that energy is lost to heat.
Yeah, absolutely. And so, yeah, that difference, you have these reactants, they have a lot of energy, and the products have less energy. Remember, energy is neither created nor destroyed. The energy has to go somewhere, and that energy is released in the form of heat and so on. And so this is an exergonic reaction. So you're always looking at the free energy, reactants, compared to the products. That's what you're looking at to tell if it's ender or exergonic. So let's look at this line. The black line here says without an enzyme. So let's not talk about enzymes yet, but notice here that what we didn't show in the bar graph yesterday is that the input of energy. So even though this is an exergonic reaction, so the reactants could be the gasoline molecule and oxygen, and then to get the end products, so the end of your combustion reaction would be like water and carbon dioxide. To get those end products and get energy released for the gasoline, you have to have that input of energy, that spark. That's what this is. This uphill here is the difference between the energy and the reactants, and the top of this hill is how much energy you need to have an input of energy, how much energy is needed to get those reactants to convert to products. So notice here it says E sub A. What do you suppose that stands for? Activation energy. It's E sub A because it's the energy of activation. All right, that's why it's called that. All right, so let's write that here, activation. So that's the energy that's needed. Once you reach that energy threshold here, then it will convert to products and you'll get your products and so on. And this is an exergonic reaction going with the gasoline example. So energy need to be put in, but then we get a lot of energy out. So how much energy comes out? Well, what happens is since you have to have an input of energy, we don't count this, the input of energy as energy being released. So therefore, as it's going down here to the products, the amount of energy released as a result of the reactants being converted to the products is still the difference between the reactants and the products. And so therefore, this is your delta G. Delta G, the reactants into the products, that's the same regardless, so that we don't count anything to do with the activation energy because that was the input, all right? And so then the output of that we don't count. And so that's what this means here. All right, so that's your delta G. Delta G is always difference between products and reactants. So then if we look at the red line, notice the shape of the graph is the same. So you still have a little bit a hill, all right, and then it gets converted to product. But the activation energy is lower. And so what that means is that it doesn't have to absorb as much energy. And so therefore, if it doesn't have to absorb as much energy, that reaction happens faster. It doesn't have to sit and wait around for more energy to be absorbed. And so, so that's what we say enzymes lower that activation energy. And so we'll talk in just a minute about how they do that. Any questions on that? Does it make sense? All right. So let's look at watch a little video here. So here's your enzyme, and so this is how an enzyme works. Notice it changed shape a little bit and converts a molecule to the products. And so this enzyme has a specific shape, and the shape determines what molecule combined here and what reaction it can speed up. So every reaction has a specific enzyme, and enzymes are very specific in what they can, what reaction they can help out with. So the reactant, some vocab here, the reactant that an enzyme acts on is called the substrate. The enzyme binds to its substrate, forming an enzyme-substrate complex. And the active site is the region on the enzyme where the substrate binds. So this part right here is the active site. And so back when I talked about sucrose being broken down into glucose and fructose and sucrase, what would be the substrate in that reaction? So back when I wrote sucrose plus water, I had sucrase here. I had glucose and fructose. What's the substrate in this reaction? It is sucrose. All right. So this enzyme, so if this is sucrase, has a specific shape. This right here would be the active site, and so sucrose can bind in here. So sucrose can bind in here. actually, by adding the water, water actually goes in here as well. And then sucrose is the substrate, and you get out of that reaction glucose and fructose, so these would be your products. So let's go through this. Yes?
Where would the water go?
So the water, like, remember, I didn't draw the shape properly here, but, so you have a ring, right? You have, these guys hooked together, two rings hooked together. And so it fits into this, and then the water comes in here, and you had an H and an OH to break those apart.
Would it hydrolyze like in?
Yes, within the active site, yes, absolutely. So let's go through an example here. I went through this a little bit when we talked about proteins originally, but we're going to just review here. And so here's the active site, and the active site has a specific shape. And notice the shape is complementary to the substrate. So now there are two substrates. I'm using this as an example. It is different than this example. The enzyme is taking a molecule and breaking it down into two monosaccharides in this case. Here, that's not going to be the case. So a lot of people think enzymes are always breaking down stuff, but that's not always the case. So when we look here, here's your active site. These guys, see how it has a complementary shape where this will fit into the active site here, and this will fit here. So that's what we're seeing here. This is called the enzyme substrate complex. We'll see in just a second, and we saw it in the animation, that the enzyme actually changes shape a little bit. I like to think of it as giving the substrates, it gives a little hug. It kind of goes around it and gives you a little hug. All right, we're going to see that that's called an induced fit. And so what happens is, it says the substrates are held in the active site by weak interactions. What is the enzyme? What macromolecule is the enzyme? Is it a carbohydrate? What is it? It's a protein, right? Chain of amino acids. So here's your chain of amino acids here. Remember, the amino acids have R groups hanging off and so on. So when we say weak interactions, these R groups are interacting. So this R group may have a negative charge, and this molecule might have a positive charge, and so they're attracted together. And so that's why it's held in the active site. And so what happens is, notice it says active site can lower the activation energy and speed up a reaction. So lowering the activation energy, let's talk about the ways in which it does that, all right? So the first thing is it can put stress on the bonds. And I'm gonna write here, to make them break easier. So this is like the sucrose. So if I'm an enzyme and sucrose comes in and binds to my active site, get in the video, sucrose comes and binds in my active site, what I can do, I hug it a little bit and put a little stress on the bonds, which then makes them break it a little bit easier, okay? And so that's what that means. The second thing is you can put together molecules. So the enzyme can not only break things down, but it can take two small molecules and put them together. And so what the second thing it could do is it can orient the molecules so they can bond together. They can. So it might put them in a situation where it orients them so that the bond that the part of the molecule that's going to react to make them bond together are touching one another. So like if we're making sucrose, so this is breaking down sucrose, there are enzymes that can take glucose and fructose and put them together, but glucose and fructose bond at a certain site. So you don't want, this is the two molecules, we don't want them like this because that's not how they're going to bond. We want them to put them together like this. And so the enzyme can help orient them so that they can bond together easier. So this is building and this is breaking down, and the enzyme, both of those cases make it so that it needs less energy for that to happen, all right, by doing one of those two different things. All right, and so here we have the enzyme converts it into products. So how we know it's products is here that yours isn't color-coded, these are color-coded, and so these are different molecules than what you started out with. And then the enzyme releases the products, And so now we have our products. Notice we had 2 substrates and we have 2 products, so we just rearranged the atoms and made the products. And notice the enzyme is the same shape as it was before. It's not used up in the reaction. So I'm going to put here that the enzyme can be reused. It doesn't change. Well, during this process, during that hugging process, it does, but then it goes back to its original shape after it lets it go. Right? And so this picture just shows you, just to illustrate, that enzymes can break things down or put things together. Because like I said, a lot of people always think enzymes are always breaking stuff down. So this is catabolism. Catabolism is breaking stuff down. So here you have, this is your what? Substrate. Where's your active site? This is the enzyme in gray. Where's your active site? The one on the left or the right? The left, because this is where the substrate is binding. We'll learn later that there are other sites that some other things can bond to. But right now, this is your active site. It binds to the active site and converts it into product. So this is breaking down, hence catabolism. Second example is anabolism. This is building. So here, your enzyme. This time we have 2 substrates, right? And the 2 substrates fit into the active site. Here's the first one, here comes the second one, and now our product is a larger molecule, so we're building it. So enzymes, some enzymes build, some enzymes break down, okay? And so this is the induced fit. This is the enzyme, what I kept on saying, hugging. Its technical term is induced fit. So the enzyme fits snugly around the substrate. I put clasping handshake. Also a little hug. All right, so here's the active site. Here's the substrate, and you can see that the enzyme is kind of going around that. And what that's going to do is either put stress on the bond so it can break down, or orient themselves so it can build. Okay, and so that is that. All right, that's where I want to stop for today. So I switched, you guys, the homework. So the homework is going to be the thing that you picked up today, and the multiple choice packet thing that you picked up yesterday. I'm going to push that back to Tuesday because we still need to do a little bit of notes for you to understand some of the questions, okay? And I'm sorry it took a little bit longer. My goal was to have you have some time to work today on that. So, like, first hour got like 15 minutes. I don't know what happened here.